Greetings of the day. I am Dr. Mithilesh Kumar Singh, presenting my research paper on topic salivary total protein and calcium in caries-active and caries-free adult. Firstly, I would like to thank my guide Dr. Gladys Rai and my co-guide Dr. G. Ratna Valigu for their continuous support and suggestion during my study period. According to the WHO, it is estimated that oral disease affect nearly 3.5 billion people, whereas untreated dental caries in permanent teeth is the most common health condition according to the global burden of the disease. With this note, I start my presentation. Introduction. The prevalence of the dental caries in the developing country is increasing at an alarming rate. Saliva possesses antimicrobial components and buffering agents which protect and maintain the oral tissue. The oral homeostasis is maintained by saliva by modulating the ecosystem within the oral cavity. Proteins that are found in the saliva such as lactoferrin, lysozyme, peroxidase, defensin and histidine can destroy and inhibit the growth of microorganisms in oral cavity. Saliva also play an important role in maintaining the integrity of the dental tissue due to the presence of calcium, which helps in remineralization of demineralized zone of enamel. Therefore, calcium acts as a defense mechanism against the dissolution of the teeth. Material and method. The study conducted on the 70 adults. These 70 adults are divided into the two groups. That is 35 adults were caries-free adults based on the DMFT score, that is the decay, missing, filled and treated tooth. Whereas 35 adults were caries-active adults based on the DMFT score. The unstimulated saliva sample were collected for the estimation of salivary total protein and calcium. For the estimation of salivary total protein, we use Biuret reagent, and for estimation of the calcium we use OCPC method, and these both the reagents are from the Urba Diagnostic, and the estimation was done on the semi-auto analyzer. Inclusion and exclusion criteria. Inclusion criteria. 18 years of age and above of either sex can consider in the study. Restoration with recurrent decay can consider in the case group. All the permanent teeth are considered, whereas in exclusion criteria, unerupted and partially erupted teeth are excluded. Primary retained teeth and patients currently on medication of the chronic disease are excluded from the study. Aesthetical analysis. Aesthetical analysis was performed by using independent simple t-test to compare mean value between the two groups that are caries-active and caries-free. A p-value of less than 0.05 considered significant at 95% confidence level. The software which we use for aesthetical analysis is SPSS version 18.0. The result after aesthetical analysis we got that in gender distribution, actually we divided into two groups, that is a caries-active and caries-free. The male in the caries-active is 13, whereas in female the caries-active are 22. In the similar way, in caries-free the male are 13 and the female are 22 with the p-value of 0.925 of gender distribution. If you talk about the total protein, which is estimated by the reagent, that is a Biuret reagent, we got the result that in caries-active the mean is 0.02 with a standard deviation of 0.01, whereas in caries-free the total protein we got the mean value of 0.03 with a standard deviation of 0.02. The p-value for the total protein is aesthetical significance, and we got 0.022. Whereas in calcium, the caries-active, the mean value is 2.63 with a standard deviation of 1.87, and the calcium in caries-free, the mean we got 0.39 plus minus 0.88 with aesthetical significance, and we got the p-value of calcium is less than 0.001. If we look over the calcium value in caries-active, we got 2.63 of mean, which is much higher than the caries-free. Discussion: the saliva, when collected with non-exogenous and pharmacological stimulation, is considered as unstimulated saliva, whereas saliva secreted by mechanical or gastro-stimuli or by pharmacological agent is considered as stimulated saliva. In 2008, a study done by Stuckey J.K. found that stimulating the flow of the saliva may alter its composition, whereas the study done in 2002 also reported that the quantity of the saliva, the concentration of some constituent, and the pH of the saliva may alter due to the salivary stimulation. Therefore, in this study, unstimulated salivary samples were collected. There are different ways for collecting unstimulated saliva, that is a draining method, spitting method, suction method, swab method. The study done by Navages stated that two best ways for collecting the whole saliva are draining and spitting method. In draining method, the saliva drip off the lower lip, whereas in spitting method, the saliva is expectorant in the container. So in this study, unstimulated saliva is collected by spitting method. In this method, we actually provide a sterile container to the patient of both the group and allow to spit in that container, and that saliva is taken in the lab for the estimation of total protein and the calcium. The proteins found in the saliva such as lactoferrin, lysozyme, peroxidase, defensin, albumin, mucin, immunoglobulin and histidine destroy or inhibit the growth of microorganisms in oral cavity. Salivary protein protect the oral cavity by protecting against wear and tear by the film of the saliva, mucin, and purine-leach glycoprotein. The salivary glycoprotein prevent the oral microorganism from adhering to the enamel and inhibiting their growth. Salivary protein also help in remineralization of enamel by binding calcium ion. Decreased demineralization by pellicle protein in concerned with the calcium and phosphate ion in the saliva. The demineralization and remineralization are taking place simultaneously to maintain the level of mineral. If demineralization is faster than the remineralization, it causes the caries in the tooth. It is also hypothesized that saliva is a blood filtrate. So, the change in the calcium level in the caries group, which we are getting, might be due to the parathyroid hormone, which play a role in maintaining its level in caries-active. Another factor for increased saliva calcium might be due to the much number of the dental caries in the patient. And it is because of the much caries, there is a much demineralization takes place, which release the calcium ion into the saliva. So the study done by the Mahajan et al. In 2017, they also found that the mean calcium level was increased in the caries affected group, whereas in a study done in 1997, they investigate 4 to 12-year-old children and found that calcium level of the saliva is higher in the children with multiple caries than in the children with a single caries. Conclusion: most of the oral health conditions are largely preventable and can be treated in their early stages. Based on the present study and available literature, it can be concluded that total protein is decreased in the saliva of the caries-active patient and calcium is increased in the saliva of the caries-active adult when compared with the caries-free adult. These are my references. Thank you.